Influence of microalgae extract on the toxicity of a carcinogenic polycyclic aromatic hydrocarbon, Benzo and pyrene in endothelial cells, by Manologov. Microalgae are eukaryotic and prokaryotic photosynthetic organisms found in freshwater and marine systems. Today, only 40,000 to 50,000 species are described, but their number is estimated at 800,000. The microalgae are mainly unicellular but can be multicellular. They are of various morphologies and size and can be mobile. The most commonly used classification is based on color. It is composed of red algae, rhodophyte, green algae, chlorophyte, brown algae, chromophyte, and blue-green algae, cyanophyte. They are increasingly used in cosmetic, in the biofuel sector, but also in animal feeds, particularly in aquaculture and in human nutrition. The interest of microalgae in the human food is due in particular to their composition in bioactive molecules. Among these components are pigments, protein, lipids, vitamins, and antioxidants. Today, only 3 microalgae are used as food algae. There is Astrospira species, commonly known as Spirula species, Chlorella species, and Endothella aurita. Astrospira species and Chlorella species are particularly interesting compared to the high concentration of protein, and Endothella aurita for its omega-3 content. Numerous in vitro and in vivo studies have shown the potential interest of microalgae extract in the prevention of various chronic diseases in humans, in particular cardiovascular disease and cancer. Pathologies also associate with the exposure of environmental pollutants, polycyclic aromatic hydrocarbon. Polycyclic aromatic hydrocarbon are environmental contaminants formed during the incomplete combustion of organic matter. They come from natural sources such as forest fires and volcanic eruptions. However, the main source is anthropogenic. They are found in cigarette smoke, in food, and in the petrochemical industry. Humans are continually exposed to these environmental pollutants, mainly by the oral route, but also by the respiratory and cutaneous routes. The pathologies associated with exposure to polycyclic aromatic hydrocarbon are cancers, lungs, bladder, liver, and skin, and cardiovascular disease. The reference polycyclic aromatic hydrocarbon is benzoylpyrene, BAP, classified as a carcinogen by the International Agency for Research on Cancer. Cellular toxic effects associated with polycyclic aromatic hydrocarbon include inflammation, apoptosis, membrane remodeling, and the release of extracellular vesicles. Your objective is to study the effects of true extract of microalgae on the toxicity induced by benzoylpyrene, and to determine if this extract can be used in chemoprevention. The 2 microalgae chosen are Ostreococcus tori (OT) and Phaleodactylum tricornitum (PT). OT is the smallest eukaryote. It is a green algae of marine origin that is known to be rich in docosahexaenoic acid and carotenoid. While PT is a diatom, also a marine origin, but rich in eocosanapatenoic acid and fucoxanthin. Both microalgae extracts were produced and supplied in tubular photobioreactor and provided by the Microalgae company. The ethanolic extract provided contain the organic molecules present in the microalgae, such as pigments and lipids. The fatty acid composition of the 2 extracts was determined by gas chromatography. According to the literature, UPT extract is rich in EPA and UOT extract is rich in DHA. The cellular model used is HMEC1 endothelial cell, known to be privileged target of polycyclic aromatic hydrocarbon. The endothelial cells were exposed to different concentrations of extract of OT or PT in the presence or absence of benzoyl during 24 hours. Cell viability, induction of bioactivation enzyme and pro-inflammatory cytokine, as well as apoptosis and extracellular vesicle production were studied. In order to determine the concentration of treatment, we studied cell viability using an MTT test on 24 hours treatment. The effect of different concentration of BAP from 0.1 to 10 micromolar and microalgae extract from 0.1 to 15 microgram per milliliter was studied. We observed a significant decrease in cell viability from 2 micromolar of BAP. So we use the concentration of 2 micromolar for the treatment with BAP. For microalgae extracts, we observe a significant decrease in cell viability from 25 microgram per milliliter of extract. We therefore choose the concentrations of 0.1, 1, and 5 microgram per milliliter. In order to verify that co-treatment, BAP and microalgae extracts, did not induce excess mortality, we compared the effect of BAP alone with co-exposed cells. We did not obtain the excess mortality with the PT extract, nor with the OT extract, at 0.1 and 1 microgram per milliliter. However, we observed a significant decrease in cell viability with the concentration of 5 microgram per milliliter for OT. We chose to keep the extract concentration at 5 microgram per milliliter because we did not observe any effect with the PT extract. We were interested in a bioactive enzyme of BAP, CYP451A1. We measure the expression level of CYP1A1 mRNA in the presence of BAP alone, extract alone, or in co-treatment. In the presence of BAP, we observe a significant increase in the expression of CYP1A1 mRNA. In the presence of PT, we observe a significant induction of CYP1A1 contrary to the OT extract. Co-treatment, BAP and PT extract also induce CYP1A1. However, with OT extract at 0.1 microgram per milliliter, we observe an inhibition of BAP-induced CYP1A1 expression. This inhibition has not been observed at concentration of 1 and 5 microgram per milliliter of OT. BAP is known to induce the expression of pro-inflammatory cytokines. We are interested in the expression of several cytokines such as TNF-alpha, IL-6, COX-2, IL-8, and IL-1 beta. In your cell models, only IL-8 and IL-1 beta were induced by BAP. We therefore focused on the expression of these two cytokines. The microalgae extract alone did not modify the expression of IL-8 and IL-1 beta. In co-treatment, the microalgae extract significantly inhibits the induction of IL-8 induced by BAP. Only the concentration of 5 microgram per milliliter ET did not significantly inhibit the expression of IL-8. In co-treatment, only 0.1 and 1 microgram per milliliter OT extract significantly inhibit BAP-induced IL-1 beta induction. BAP is known to induce apoptosis. We verified that BAP induces apoptosis in your experimental collection. In your cell model, BAP induces apoptosis from 0.1 micromolar. We also verified that the different concentration of microalgae extract did not induce apoptosis. In your experimental condition, no concentration of the two microalgae extract used induced apoptosis. We therefore looked at the effect of co-treatments on apoptosis. The PT extract did not modify the apoptosis induced by the BAP, unlike OT, at 0.1 and 5 microgram per milliliter, which inhibits apoptosis induced by BAP. The OT extract at 0.1 microgram per milliliter seems to be more interesting to find against the harmful effects of BAP. So we decided to focus on the extract. We were interested in the production of extracellular vesicles. Extracellular vesicles are membrane nanostructures formed by all types of cells found in biological fluids. Their main function is intracellular communication. The number and the composition of the extracellular vesicle are markers of the physiological and physiopathological state of the producing cells. We studied the effect of BAP and OT extract at 0.1 microgram per milliliter on extracellular vesicle production by nanoparticle tracking analysis (NTA), a technique that allows us to determine the number and size of vesicles. We observed that BAP stimulates the production of extracellular vesicles in an endothelial cell. In the presence of the OT extract at 0.1 microgram per milliliter, we did not observe any modification in the number of vesicles. In co-treatment, we observed an inhibition of the production of extracellular vesicles induced by BAP. In conclusion, OT extract inhibits the expression of BAP-induced inflammatory cytokine IL-8 and IL-1 beta, as well as apoptosis, the expression of a BAP bioactivation enzyme CYP1A1, and inhibits the production of extracellular vesicles. The PT extract inhibits BAP-induced pro-inflammatory cytokine IL-8 expression. The OT extract therefore seems to be interesting for use in chemoprevention of the toxicity of BAP. Extract containing different bioactive molecules. It's difficult to associate the effect observed with the activation of a single signaling pathway. However, much of the toxic effect of BAP, induction of CYP1A1 and apoptosis, is associated with activation of the aryl hydrocarbon receptor, AHA. BAP, by activating AHA, has the ability to stimulate its own metabolism by inducing genes involved in the metabolism of xenobiotic, such as CYP1A1. AHA can also interact with the NF-kappa B factor and induce the expression of cytokines such as IL-8. It is known that DHA is able to activate the NF2 pathway involved in the antioxidant response. Since OT is rich in DHA, it would be interesting to determine the implication on NF2 pathway. In addition, in order to determine whether the effect observed on the toxicity of BAP is due to the composition of DHA of OT or a synergic effect of the molecules present in the extract, it will be interesting to study the effect with only DHA. This work was supported by the Programme Environnement Santé Travail of ANSES with the funding from IGMO Cancer in the context of the Cancer Plan and by the collectivity locale Mayonnaise. Thank you for your attention.